Um, Great. Well, can everyone hear me okay? Okay. And thank you, Indica, for inviting me. I was saying earlier, I haven't been back to Ann Arbor. I grew up here, but I haven't been back in about 10 years. So it's really cool to come back and kind of see, some things are familiar, some things not so familiar, but, to kind of visit what I've thought of as home for a very long time. So it's very cool. And I'm also going to tell you today about things that we've been working on in my lab at Salk. Our lab is really interested, as Indica said, in terms of 3D genome structure and sort of how our genome is organized in space. And I'm going to kind of present some data that we've been working on, really thinking about how our studies of 3D genome structure, I think, are helping us to think about sort of the impact of particular classes of sequence variants. And this would be both structural variants as well as somatic germ-line single nucleotide polymorphism. Okay. So I guess I'm just going to start giving you a kind of a brief outline of what I'm actually going to talk about today. So for the first part, I'm going to give a little bit of intro to generally introduce why we're interested in how your genome folds up in space, and introduce some of the tools that we use and some of the general principles we observe. I'm also going to talk a little bit about, in the first part, a story about what happens to a particular feature of genome organization we call topological domains. These are also called topologically associating domains, or TADs. And so what we can see that basically happens to these in the context of structural variation in cancer genomes. For the second part of the talk, I'm actually going to talk about some new method development we've been working on, trying to do basically single cell profiling using both 3D genome structure as well as DNA methylation, and trying to use this in complex tissues with really kind of the goal of this, at least one potential long-term goal, to try to understand variants that might be associated with particular human diseases. So what are we talking about in terms of 3D genome organization? So we have some 3 billion base pairs in a haploid copy of the human genome. These all have to be compacted down within the nucleus. And I think most people are sort of typically thinking about DNA in sort of a linear fashion, as sort of a sequence of nucleic acids that we might be able to layer some sort of feature information on top of, right? So we can annotate genes or regulatory sequences. I would say this is kind of the conventional way that people think about sort of DNA, right? But we actually know that this is really actually folded up in three-dimensional space inside of cells, so that the different genes and regulatory sequences come in close contact, and that this can really impact sort of genome function. So this is kind of a major, question that we're interested in. And so one kind of, just as an example, one reason why one might be interested in 3D genome organization is sort of given in sort of a toy example here, which is that as a field, the field of genomics, we've actually gotten very good at mapping regulatory sequences in the genome using things like ChIP-seq, ATAC-seq, whatnot. And if you do this for your favorite transcription factor and your favorite sample, you'll inevitably come to a situation that looks like this, where you find some enhancer that sits here, and maybe there's 2 genes nearby. And sort of from the one-dimensional sort of a priori sequence here, you don't know what this enhancer is doing. You don't know if it regulates gene A or gene B. But we also know that the way that enhancers work, or at least the sort of dominant model for how they work, is that they're brought in close spatial proximity with the genes that they regulate. And this allows proteins that bind to the enhancer to interact with proteins that bind at the promoter, and this facilitates transcription. And so in this sense, we can begin to say, maybe map which enhancers are in proximity with which genes to get better ideas of what are the target genes of regulatory sequences. And so you can think about this problem in a slightly different way, which is that if you then say layer some sort of genetic information on top of this, say there's a polymorphism that sits within this enhancer, and perhaps we know this polymorphism may be associated with a particular trait or a human disease, this actually kind of reformulates this problem slightly, which is to say that now the question is, if this enhancer targets gene A or gene B, this allows you to make insights in terms of sort of the molecular pathogenesis of this variant. In other words, saying, if this enhancer targets gene A, you can put that in one particular pathway or another, and this gives you insights in terms of sort of the disease biology, as well as potential sort of therapeutic personalized medicine approaches that might be amenable for treatment. The other aspect in which I think we're thinking about this problem a lot is in cancer genomes, and then also in some cases in the germline, you can have mutations that will basically break a chromosome and stitch it back together with a new partner, right? So we would call this a structural variant. And in this case, the problem again changes to say, well, if this, does this enhancer now regulate this gene that's now in a novel context, right? And this is particularly important, say, if this gene is an oncogene. Now it becomes a question of if this is really regulating this gene, this might be part of what's driving oncogenesis. So in terms of the tools we actually use to kind of map out these structures, so the dominant tool that we use in the lab is it's sort of a molecular biology-based assay called Hi-C. This was originally developed by Job Decker and Ayers Lieberman Aiden and Eric Lander as well. And the idea is that you take a population of cells and you fix them with formaldehyde, such that if you had an enhancer and promoter that have been brought close together in spatial 3D space, these will actually become covalently linked to each other because of the cross-links. You then digest ends of DNA with the restriction enzyme, and then you can fill these in with nucleotides, one of which is linked to a biotin moiety. You can then ligate these ends together, and this preferentially occurs between things that were originally in close three-dimensional space. This generates a contiguous piece of DNA that doesn't exist naturally anywhere in the genome and is now conveniently marked with a biotin. We can then purify DNA and isolate these ligated fragments using a stripped additive-coated bead, and then subject this to high-throughput paired-end DNA sequencing. And this gives you a picture of basically all the possible interactions that might be occurring in a population of cells at any one time. In terms of how we actually look at the data, I'm sure there are people here who are very familiar with this, but just in case there are not, what we're really doing is we're basically taking these reads and then we're aligning to the genome and we're asking how different regions contact each other. And the way this really looks is we start by taking the one-dimensional sequence and we break it up into bins. And these can be of sort of arbitrary size. It depends on the resolution that you're looking at. For low resolution data, these might be 1 million base pairs in size. For high resolution data, these would be about 1 kilobase in size. Oh, hello. Yeah, right? What we are essentially doing is mapping the reads that go between bins, right? So in this case, there are 5 reads between this bin on the right and this one on the left, or in the one in the middle. And we can assign that a particular color. And then we can say there's 3 between these 2 left-hand bins. We assign that a color. We say there's 1 between these 2 outside bins. We assign that a color. And this way, we build a color-coded map of how these different regions of the genomes were contacting each other, such that some of the real data looks like this. This is from this PHC1 gene locus. So we could say, how does maybe this promoter proximal region interact with this upstream enhancer by finding where the 2 intersect off the diagonal and looking at what signals are there. So in this case, this kind of pink signal suggests these interact with some level of frequency, whereas white would suggest they interact infrequently, if at all. And so we've been doing these assays now for several years. And one of the features that we were really able to identify now many years ago is what we call topological domains. They're also called topologically associating domains, or TADs. And really the way that we identify these is we just start by looking at the data. And so this is Hi-C data from neural progenitors over about an 8 million base pair stretch of the genome. I think one of the things that you can almost immediately appreciate is there are these little triangles that pop up, right? So triangle, 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 et cetera. And one of the things that we thought was interesting about this is if you think about what a triangle would mean in this kind of data, basically this stretch forming a triangle is a sequence that interacts a lot with itself. This stretch is triangle is a stretch that interacts a lot with itself. But the relative lack of signal going between them suggests that they're at least relatively spatially isolated from each other. And so we can kind of represent this in cartoon form. And again, this is really just a sort of a cartoonish model to conceptualize this. Is something like this, where one cluster might correspond to one triangle here. Second one corresponds to this triangle. The other thing that we were able to see that I think is actually pretty remarkable is when we were able to profile these kind of patterns across different cell types, we were actually able to see that the position of where these kind of domains were occurring in the genome was actually very stable across what are fairly diverse cell types. We basically think that these probably are forming some fundamental unit of how your genome is being organized. And this slide is really to summarize work that's been done by many labs at this point over now many years, which is to say, what do we think these really do? So the idea is these are maybe 200 kilobases to 2 megabases in size. They seem to form as a result of kind of an interplay between this transcription factor called CTCF and a complex called cohesin. The dominant model, I would say, in the field right now is this model called loop extrusion, which is that these are facilitated by basically DNA looping events. And in terms of function, I think the most clearly described function that we have at this point has to do with these regulating enhancer promoter communication. And so some kind of evidence to support this is particular cases where we know a particular enhancer regulates a specific gene, say by genetic evidence or whatnot. These are almost always occurring within the same topological domain. There's a trend towards gene expression being correlated within TADs across different tissue types. If you take reporter constructs that basically have minimal promoters and you insert them in the genome, they'll start picking up expression patterns of nearby enhancers, but only those that are within the same domain. And from, I think, really seminal work from Stefan Munlos's group, they were actually showed that structural variants that would alter the TAD boundaries would lead to altered enhancer promoter communication and altered gene expression in these human developmental phenotypes. And his last point was really something that I thought was super cool when we were starting my group at Salk, which is if you kind of compare some of the things I've told you, right? So structural variants would be things like these inversions, deletions, insertions, or translocations, where you're taking a chromosome, you're breaking it apart, and you're stitching it back together. If you kind of contrast this with this really stable pattern in terms of where we were seeing these domains, seemingly these would have to have a pretty big effect in terms of how your gene was being organized. And we wanted to get really a better idea of how this would be occurring and what the consequences are of these kind of events on 3D genome structure. And so I would say also, this is not necessarily like a purely academic problem in that structural variation is actually extremely common in human cancers. So this is data from a cohort of 500-something breast cancer patients where the median tumor has about 85 rearrangements. So that's a very kind of rich landscape to alter these sort of 3D genome structures. And we know that rearrangements that basically alter enhancer promoter communication are basically the causal defining genetic lesions that contribute to a variety of different human malignancies. This is probably best described within hematopoietic cancers. So things like these T-cell leukemias with these deletions near TAL1, immunoglobulin rearrangements near MYC in the particular classes of lymphomas. And there's additional cases of this that are basically being identified all the time now. And so we thought that this was particularly interesting and basically wanted to kind of dive into this a little bit more to say, can we do this in more systematic ways of identifying TADs that are rearranged in cancer and understanding the consequences a little bit better. So I'm going to kind of go through this little bit very quickly because I know that Feng Yue came like a year ago and talked a little bit about some of this. And so we had collaborated with Feng to actually try to use Hi-C data to find rearrangements. The basic way that this works is if you take Hi-C data, this is from basically normal, non-rearranged portions of the genome, where this is interactions within, in this case, chromosome 1, whereas these are interactions between chromosome 1 and 2. And we compare that with data from regions of the genome where we know there's something like a translocation. You see these sort of really dramatic alterations in signal, right? So this kind of diffuse hazy pattern you see between two non-rearranged chromosomes is very strongly altered at this specific point, and the very kind of dramatic transition in contact frequency right at the breakpoint of where we know this translocation occurs. And so we, teaming up with Feng, we actually developed some computational tools to go in and find this kind of signature and identify regions of the genome where this occurs. And so subsequently, we're able to use that data to then kind of ask some sort of basic questions about what's happening when we find these rearrangements. So this is basically an example case where we find a translocation, in this case between chromosome 9 and 18, and where we think this is basically the Hi-C data within the region on chromosome 18. This is the Hi-C data within chromosome 9. And because we have the Hi-C and other data like bionano or whole genome sequencing, we could see that these were really actually fused together now into a new translocated allele. And then we can overlay Hi-C data that crosses the breakpoint, and you can see them form this kind of nice little triangle here, right? So we think this is basically, this translocation is creating to the formation of a new domain. We've been able to do this a little bit more systematically to ask, is this kind of a general pattern? And I think the answer is yes. So basically using a couple different approaches, both in the paper we published with Feng, as well as in something that's been a collaboration with Andy Futrial's lab at MD Anderson. What we think the general trend is kind of illustrated by this kind of diagram here, is that when you get a structural variant, what you're basically doing is taking the new nearest TAD boundaries and stitching them together to form a new domain across the breakpoint. And that seems to be the kind of general rule that we're observing in the data. Yeah, I mean, there's some way that these have been called neo-TADs or fusion TADs. But yeah, so it's basically a novel TAD, novel regulatory environment, we would say. So from the standpoint of like a pure aneuploidy, like just gaining one additional copy, I don't honestly know. I've seen some data from like Down syndrome that it doesn't seem to have a huge effect of just gaining one additional copy. But in this case, this is not just a gain potential of copy, it's also a juxtaposition. So I think what's driving this is primarily the novel juxtaposition of genetic sequences in this case. I think it's an interesting question. Like if you stuff, like a genome doubling event happens all the time in cancer. That's twice as many chromosomes. Seemingly that would do something, but I don't have a particularly good answer for that. Yeah, so basically we were able to see these kind of patterns, and we thought this was interesting, but we had a few questions that we wanted to address. So one is being able to identify these. We said, well, are there regions that we can find where there's recurrent alterations to TADs in the genome, in cancer genomes? And two, in the event that these were forming, is there anything in particular that's actually required for them to form or to contribute to sort of oncogene or gene expression changes or gene dysregulation? And so the way that we basically have tried to go about doing this is by profiling sort of many different cell types at this point. So we've either generated or analyzed high-C data and now from 92 different cancer cell samples. So this is a mixture of both patient samples as well as cancer cell lines that we have in the lab. About a third of this is data that's already publicly available, so we just download that and reanalyze it, and the rest is stuff that we've generated. And we can then take these tools that we developed for finding structural variants and apply that in this context. And so this is actually an example of one that we find in a breast cancer patient sample. And so our tool, we can basically pull this out and say, here's a translocation, and then we can zoom in and identify the breakpoint in pretty high resolution. And so we've basically taken this tool and we've applied this to these samples. We've also included a set of like known rearrangements, so tumors that have kind of disease-defining translocations. So these are ones from these mixed lineage leukemia tumors where they all have a mixed lineage leukemia or KMT2A translocation, and that's to different partner regions. And so in this case, it's AFF1, AFDN, and in this case, the MLL-T3. And so we can basically find these kind of known events in all cases. So we actually think that it's pretty sensitive for finding these large-scale structural changes. So now we can basically apply this across this whole system and say, where do we find change? And so this is basically kind of a Manhattan plot style pileup of all the regions that we find altered TAD structure as a result of these structural variants. And we develop a fairly simple statistical model to account for the size of the TAD as well as the overall frequency to say, are there sites that we can find that are being recurrently affected? And the answer is yes. There's actually 12 different regions that we find in our data that show evidence of recurrent TAD structural alterations. And at least some of the top hits are sort of your classic, oncogene loci that we can see. So the most frequently arranged site is MYC. We can see frequent events at telomerase and cyclin D1. Some of the other ones that I don't have labeled here, it's partly because there might be many genes or multiple oncogenes per locus. We don't necessarily know if there's one target or multiple. Well, we'll get to it. Yeah. So we thought this was cool, but I think, one issue, right, is we wanted to be able to say, can you see this in independent data set, right? So we have a lot of cell line data, and you could argue, well, maybe there's some weird cell line artifacts. And so what we've been doing is looking in data that's been generated through this International Cancer Genome Consortium. And so this is whole genome sequencing data. They actually have this now from like 2,000 patients where they have matched tumor and germ-line whole genome sequencing, and they have fairly high quality uniform structural variant call sets. And so if we perform kind of a similar analysis, in this case over 3,000 TADs across this whole data set, we can see the sites that we identified in our Hi-C data are labeled in red. And so you can see that some of these are still very highly rearranged in the ICGC data, whereas a few of them are really not particularly enriched and maybe kind of false positives in our data. And I should also say the way that we do this, right, we don't have Hi-C data from these 2,000 people. So we're using kind of a crude proxy for a rearrangement between TADs by basically saying if you have a structural variant where one break end maps in one domain and the other break maps in the other, we're sort of assuming this has to alter the TAD structure somehow. So we're basically calling these kind of inter-TAD rearrangements. And this is basically the frequency of inter-TAD rearrangements by locus. And again, you can actually see that MYC is the most frequently affected site, again, in a genome here. And because of this, we wanted to focus a little bit more on MYC and try to say what's going on here and what might be interesting. We could see that actually these MYC inter-TAD rearrangements in this data were actually occurring in a wide variety of different tumor types. So MYC rearrangements in terms of these regulatory fusion alterations are actually very well known to occur in lymphoma samples. But we're seeing these in really a wide variety of tumors. So the most frequently rearranged subset is these, blinking on the word, endometrial cancers. So endometrial cancer, ovarian, gastric, ovarian, this is a lymphoma subset, esophageal, breast cancer, lung cancer. So we see these really all over the place in all the different tumor types that are in there. There's some that are at lower frequency, and it's not totally clear what that is, but we're really seeing these all over the place. And we can actually see the same thing if we look in our Hi-C data, which is this is some of the fusion events that we find at the MYC locus. And so we can find, here's MYC, and then you get these novel signals that form, in this case, with what's chromosome 2, and this is in a breast cancer cell line, another breast cancer, get the context there. Pancreatic cancer sample, you see some novel contacts here. And this is in a lymphoma where these events are actually known to occur, but you can also see these kind of novel contacts here. So the other thing that's cool about looking in the ICGC data is they have RNA-seq data for not all of the 2,000-something patients, but a lot. And so we can ask, do these also show elevated MYC expression? And so what we can see is looking across all tumor types, we do see that there's an elevated expression of MYC in tumors that have these kind of inter-TAD rearrangements at the MYC domain compared to ones that don't. And we can see that this also occurs within specific subtypes. Again, malignant lymphoma is a subtype where this is known to occur, but we can also see this in other subtypes like breast cancer. And so MYC is actually, a very well-known oncogene. It's known to be amplified in many different kinds of cancers. But we don't actually think that this is a product of copy number changes. And we think that we can see this because if you filter this pan-cancer data for tumors that lack any kind of high-level copy number change, you still see that the ones that have inter-TAD rearrangements show expression. And if we're even more strict and we say we restrict to only having tumors that show no copy number change at all at the MYC locus, we can still see that these tumors that have inter-TAD rearrangements show elevated MYC. Necessarily strictly due to copy number changes. I think at least a reasonable component of this effect is actually being due to altering sort of the regulatory architecture of the gene. So the other thing that we looked at is sort of partner regions, and this is actually where it gets to be a little bit confusing, at least initially. So in lymphomas, it's very clear. There's MYC and there's IGH, and they form a partner, and that is basically a defined event. And you can genetically look for that, and that tells you something immediately about the patient. If we ask in these pan-cancer data, is there recurrent partners? The answer is basically no. You can see there's maybe some chromosomes where there might be a slight enrichment on a particular arm, but for the most part, these go everywhere, right? And I think coupling that with the fact that in the expression data, there's still a fairly strong overlap between the non-rearranged and the rearranged tumors. We thought that this was kind of interesting to say, well, given that you see this heterogeneity of partners, as well as this sort of, enriched but not an absolute difference in expression. One question was, well, how do we tell a tumor that's actually being driven by a regulatory rearrangement? We can't really do it by saying, well, if it has X partner, it is by definition activating MYC expression. And we can't necessarily assume that if it has just any rearrangement that it has elevated expression. And so we wanted to try to address this. And the way that we've gone about trying to do this is actually by doing CRISPR-Cas9 genome engineering in particular cell lines. And so we've been doing this in neuroblastoma cell lines, basically trying to engineer novel translocations. The way that this works is part of the reason why we chose neuroblastoma is they have this really interesting phenomenon where there are some neuroblastoma tumors that have high-level nmic amplifications and express high levels of nmic. And there are some neuroblastomas that have rearrangements at cmic and express high levels of cmic. And when I say mic in these cases, I'm talking about the cmic gene, not the nmic gene. And so we had data where we could say this is a cell line that expresses very high levels of cmic, and we have a translocation here on chromosome 8 at the mic locus. Whereas this is a cell line that doesn't express much cmic at all and has no rearrangements at the cmic locus, and so it's basically kind of a blank slate. And so we said, let's take one of these cell lines that doesn't express cmic, and let's try to just screw up the locus and see what kind of expression changes we can make, and if we can make any sense of the consequences. And this is the RNA-seq data for these two cells. So what we've been doing is we basically take two guide RNAs, we label them with different plasmids, we transfect them into cells, plate them as single cells, and grow up colonies. And then we can actually screen through the colonies. It's actually fairly easy to do the screening because we're looking for rearrangement just by PCR. If it's there, there's a band. If there's not, there's nothing. It's inefficient, but it's an easy screen to do. And we can then do, say, PCR across the Sanger sequencing across the PCR breakpoint to confirm the rearrangement. And we can also do things like chromosome painting to actually show that the engineered cell lines have these translocations, in this case, in chromosome 8 and 7. So the other thing that I should say is, we can also confirm that these events, we can see rearrangements by Hi-C as well. So these are 4 different clones where we engineered rearrangements to the same locus. And in each of these cases, you see these novel contacts crossing the breakpoint. The other thing I should say is we actually specifically were sort of aiming to target regions where we thought they were super enhanced. And the reason for this, and I guess to give a quick background, super enhancers, the idea is they're a class of regulatory sequences that are basically just the strongest enhancers in the genome. They tend to have a sort of exponentially higher activity than sort of a typical enhancer. And they were, described by Rick Young, as well as I think some others historically, as basically these large clustered enhancer regions in the genome. And there's some pretty clear data that these are generally associated or found near critical lineage developmental regulatory genes. And what we could actually see is in our Hi-C data, we could see that in the TADs that were forming fusions, basically globally, not just at MIC, but everywhere, you could actually see they were more likely to contain super enhancers than those that we wouldn't find any kind of TAD-like rearrangement. We thought, well, maybe this is interesting and we should include or focus a little bit on super enhancers for this kind of analysis. So we've been doing this. We've chosen now sort of a handful of different regions across the genome. So MIC is here on chromosome 8. And we have 1, 2, 3, 4, 5, 6, 7 sites that were engineered to, in this case, we have replicates from different sites. And then we've basically done all of the screening and confirmed all of these. And then we can ask, okay, what kind of gene expression changes do we see? And so on the left, these first 2 columns are replicates from the cell line that naturally carries this MIC translocation. These are the parent cell line that normally don't have translocation and don't express MIC. And then from here on are all of the clones that we've done where we've engineered rearrangement of different loci in the genome. And I think what you can see, which is kind of nice, given that we saw this heterogeneity in expression in the ICGC data, is we actually could see that there's a fairly broad degree of heterogeneity in expression here as well. So some of the clones express very, very, very little amounts of MIC, and others express pretty high levels. And there's actually almost, I think, a 500-fold difference between the most induced and the least induced clone. And so we can go back to this kind of super enhancer model and say, well, does this explain everything? And the answer is not exactly, but kind of. So we could see that in clones where the breakpoint contained a super enhancer, they generally had higher levels of expression. I say that sort of, but not always, because calling super enhancers is not necessarily a definitive. There's only one way to do it, and your ability to call them depends on whatever parameters you choose. And so in this case, we're basically saying, let's try a variety of different parameters to call these. And so the sites where we sort of reproducibly call them, at least relatively so, regardless of parameters, we generally tend to see slightly higher expression. But again, this is a little bit kind of hand-waving, and so we wanted to say, are there better things that saying, well, super enhancers kind of explain this? And so what we could actually see if we look in particular cases are these interesting examples where this is two different events. And so there's a little bit of signal right there. And they're two different loci. So this is on chromosome 12, this is on chromosome 1. And they're about the same size. The partner region is about 1 million base pairs. They look otherwise very similar. But if we look at the gene expression, this one expresses fairly high levels of MYC, whereas this one doesn't induce it much at all. And we said, what's the difference here? And what it really comes down to is if you look at the partner region, so this is this 1 megabase stretch on chromosome 1, there's sort of a lot of enhancer signal there, right? So this is H3K27 acetylation chip-seq. If you look at this region, there's some, but not a whole lot, right? So we think it's really a product of what we're calling sort of an enhancer load in the partner region. And this was kind of reminiscent of a recent story that was actually a preprint from Jesse Ingritz's group, where they developed these models that they call activity by contact. The idea is that through these high throughput CRISPR screens, they have this kind of relatively simple model for saying, does an enhancer regulate a gene? And it's essentially the likelihood of this is, or the marginal effect is the activity of the enhancer times the contact frequency with the gene that it's in proximity to. And so we can actually apply this kind of same model. In this case, we're not just trying to predict the marginal effect of any enhancer, we're actually trying to predict the total level of expression of MYC. And again, this is basically a weighted sum of the activity of the enhancers, but weighted by the contact frequency with the MYC gene itself. And this is basically the best way that we can find to predict the MYC expression in the context of these rearrangements. And we've done some sort of alternative things. We're saying, what about the closest super enhancer? Or you just sum enhancers, you don't weight them by contact. And these work. This explains it to an extent, but it's just not as good as the activity by contact. So we think this is kind of the most likely feature or way of explaining this heterogeneity that we're seeing in MYC expression. Yeah, is right here. So the other thing that I think is cool, and this is really thinking forward in terms of saying, can we expand this kind of result to other oncogenes or other genes in general in the genome, is if we ask, well, what about other genes at the MYC locus? Are there other genes that we find that are dysregulated? And the answer is basically no. So MYC is in a relative gene desert. And there's not that many other genes that are pretty close by. But if we compare, say, MYC, we have some clones that have very minimal level of activation that I'm basically calling negative, and some that really activate that I'm calling positive. You really don't see any other similar kind of pattern across any of the other genes at the locus. So there's something about the MYC gene itself that I think is actually particularly susceptible to these kind of rearrangements. And I think it may, these activity by contact models I think are really cool, but we may have to be smart in terms of saying what genes are actually affected by this kind of stuff. So that is the first part of the talk. How am I doing on time? Okay, doing okay. So I guess just to kind of summarize, we like TADs. We think that they're a fundamental feature of genome organization. We think that we can see evidence that they're forming as a result of structural variants. You form new domains. We can find evidence of recurrent events. And we think that MYC is really one of the sites that's most affected. There's a couple of additional lines on here that I didn't really talk about, which is that not all structural variants necessarily are activating MYC. I think this goes back to this expression heterogeneity that we can see, and that really there's this super enhancer effect as well as this kind of activity by contact consequence. And so now I'll go on to the second part of the talk, which is really a method development. This is something we've been doing in collaboration with Joe Ecker's lab at Salk. Which is a method development for basically trying to do better profiling of 3D genome structure in actual patient samples or tumor samples or tissue samples from real individuals that are complex heterogeneous mixtures. And again, I think one of the major goals of this, kind of tying into this idea of thinking about sequence variants, is actually to use this to try to better understand things like a GWAS linked variant. In terms of what the target genes are, what the relevant cell types are. Yeah, so I guess that's going back to here. So like I said in the introduction, one reason why people are interested in sort of mapping enhancer promoter connections is to try to better learn what are the target genes of some variant that we think might disrupt this enhancer function, right? And this is not just kind of a theoretical thing. There's actually clearly ongoing sort of human genetics GWAS literature. And there's kind of one cool example from the last several years where This is a GWAS, Manhattan-style plot for regions that are linked to obesity. And the top hit in basically every obesity GWAS study ever is this FTO locus. And what's interesting is that when people have subsequently done these kind of mapping to try to identify target genes, what you see is there's this cluster of SNPs that are in linkage disequilibrium that are associated with this obesity phenotype. And then if you map by high C, they're right here. They actually seem to form fairly long-range contacts with this IRX5 and then also with this IRX3 gene, right? And so that if you actually look in terms of RNA-seq data, classifying patients by either non-risk or risk genotypes, you actually see that IRX3 and IRX5 are the only genes that really show an expression difference in patients based on classifying them by risk genotype. So in this case, I think what this is really saying is that this variant, even though we say it's within the intron of this FTO gene, is really likely regulating these genes that are several hundred kilobases away. And so I think this is really cool. And I think that there's more and more of this that's appearing in sort of the human genetics literature of using high-C data to try to understand target genes of GWAS polymorphism. But I think there's some challenges. And I think the challenges are really on the end of the people who generate the data. And this is kind of an example case, which this is a recent GWAS study in Alzheimer's disease. And they have identify, validate a handful of loci, identify some new ones. And then they have this thing about saying, we're going to try to link these to target genes using chromatin contact data. And because of the association of Alzheimer's with microglia, which is an immune cell type in the brain, they say they used spleen and liver high-C data to do this. Yeah, so this is obviously not microglia. But I think, I really don't mean to diminish their study. I think this is very cool, but they're basically using the data that's available, right? So I honestly think this is more a challenge for people like me who generate this kind of data. Like, can we actually generate this in the relevant cell types? And so the question then is, well, okay, microglia, that's a very small cell type in the brain. How do we actually do this, right? And so I think what is the ideal approach, right, is to actually try to develop methods to identify chromatin interactions in these non-coding variants. And basically, what are the relevant cell types for disease function in vivo, right? And so there's obviously many ways you could do this. You could use, let's say if you had a mouse, you could tag some cell with a reporter. But obviously, that's going to be specific to mice. You can't do that in humans. You could, in theory, have an antibody to sort some sort of cell surface marker, but that's not necessarily available for every different cell type, and it's also not necessarily going to get you a particularly pure population, depending upon the cell type and the marker. And so one way that sort of the single-cell genomics world has been able to kind of get around this problem is to say that you basically take a complex tissue, you perform some sort of single-cell experiment. In this case, this is drop-seq, so single-cell RNA sequencing, where each of these cells in this complex mixture is now labeled with some sort of barcode. You measure the RNA label, you know the barcode, and you can use, your favorite t-SNE plot to then identify single cells that look like each other, do some sort of clustering, and then say, well, now we have information from each of these constituent cell types based on sort of a in silico purification in this case. And so we said, well, in theory, right, you could do the same kind of thing with high-C data. People have done this with other data types as well. So this is from our collaborator, Joe Ecker's lab, where they've been able to show you can do this with DNA methylation to generate cell type specific single cell profiles to resolve cell types, in this case, in the human brain. But one of the challenges with doing this with high-C data is there's sort of intrinsic noise in the system. And we've actually known this for a very long time. But this is a really kind of nice example of it from a recent paper from Tom Mistelli's lab, where these are basically all, I think, mouse embryonic stem cells. And they're labeling the same loci in each case. And what you can see is, like, if you look in this cell, the blue and the red are right next to each other, and the yellow is far away. In this cell, all three of them are on top of each other. And in this cell, they're all over the place. Nobody's next to each other, right? So this is basically spatial genome organization within what should otherwise be a homogeneous population of cells. And there's obviously a tremendous amount of variability. So if you use this to try to do these kind of single cell clustering algorithms, this intrinsic heterogeneity is basically noise that you're throwing on the system. And so we were concerned, or at least we didn't necessarily think that this would be particularly robust to identify particularly related cell types. And so what we did is kind of borrowing from this idea right here that you can do the clustering based on DNA methylation. We said, let's try to develop an assay where we measure both of these things at the same time, right? So what we're doing is we're calling this method multi-omic profiling of basically DNA methylation and 3D genome structure, as we call it, single nucleus methyl 3C sequencing, so SNM3C-seq. And the way that this really works is you basically, the initial steps are just like in a normal high-C experiment. You digest, we actually omit this fill-in step for sort of technical reasons that if you're interested, we can talk about later. And so we basically just do a ligation. This is all occurring within an intact nucleus. So you sort nuclei into 384-row plates. And then you can take this basically through a protocol for doing single-cell bisulfite sequencing that was developed in Joe Eckert's lab. And then at the end, you get barcoded data from each cell. You can sequence this on a sequencer and get, in this case, both 3D genome and methylation data from the same cell. So we do some controls. Basically, we say, if we generate this now in a bunch of different single cells, we get very sparse contact maps from any individual cell. We get very sparse DNA methylation profiles from any individual cell. This is pretty typical for single cell data. If we pool them in bulk, we can actually recapitulate the same kind of patterns you see in bulk IC data, right? And then we can actually see some patterns that we think are cell type specific. So this is for mouse ES cells. They have this kind of small tad at the SOX2 locus that's actually lost in cells that are not ES cells. So it's specific to ES cells. And when we profile this in ES cells, we think, the data is kind of noisy in this case, but we think there's still this kind of small tad there. And we can also see that it recapitulates the DNA methylation patterns that are observed, in this case, comparing at the DPPA4 and 2 locus. And so we've been able to do this, and we can now apply this in particular settings. So one, basically doing this as control, is we apply this to 2 different mouse cell lines, embryonic stem cells and this mammary gland cell line, to basically say, can we use this? Can you actually distinguish the 2 cell types from each other? And so if we use the DNA methylation data, we can very clearly and easily separate the mammary gland from the mouse ES cell data. If we're using contacts, we can still do this, but it actually requires a little bit of work. We have to kind of play around with the conditions in order to identify it. And so at the end of the day, I really think the methylation is a considerably more robust way of partitioning these cell types than the contacts data. I'll show you another example of this in a minute. Yes. Yes. Well, so that's the thing. The dominant component here is not separating by cell type either. This first principal component is actually coverage. So clearly the coverage, like basically you separate cells better at higher coverage. This is negatively correlated with coverage. Whereas at low coverage, you don't. So I think that the issue is, if we had super high coverage, maybe this works as well as DNA methylation. But, the coverage is the same in terms of the genome in both cells. Either it's the same data, right? So I think what it really comes down to is the partitioning in terms of the sparsity of the data is just better with DNA methylation compared to this. So the thing that we've done, that we've kind of applied this in a cool setting, is we've basically applied the sample now to actual human brain tissue. So this is from postmortem individuals, prefrontal cortex. We basically put it through the protocol, and we include a sorting step to bias for the generation of neurons. I didn't actually know this before we started, but the dominant cell type in the human brain is not neurons. It's basically glial cells, which I didn't know. I'm not a neuroscientist. So, but yeah, so we bias for neurons, and then we can apply this through the method. And then we, as everyone else does in a single cell experiment, we generate some T-SNE plots. And so we can do this and now begin to classify different cell types. And there's a couple cool things that I think about this. So this is doing the clustering based on CG methylation, which is sort of the classical methyl context in terms of a CpG dinucleotide. You can actually distinguish all these different cells particularly well, or fairly well when you do this. You can also do this with non-CG methylation. This is actually rare in most cells, but it's actually fairly highly present in the brain as well as in heart tissue. And you can actually see that Non-CG methylation doesn't actually distinguish glial cells like oligodendrocytes or microglia, but it does a very good job of distinguishing neurons from each other. And then we do the same thing for high C. What's interesting is the high C data can actually distinguish some of the non-neuronal cell types. So it does a decent job of distinguishing oligodendrocytes, astrocytes, endothelial cells. But we get this giant ball cluster here of neurons, right? So it doesn't do a particular job of distinguishing, which I think is kind of reassuring because it's like, well, that was the whole point of doing this in the first place, is to try to see if we could use this to distinguish potentially related cell types. And there is some structure there. It's like this half tends to be excitatory and this half tends to be inhibitory, but they're within the subtypes, they're really still all clustered, mixed together. And so then, I guess this is a couple of examples. So the part of the way that you do the cell type classification is you're basically looking for hypomethylation over either gene bodies or gene promoters, because we obviously don't have RNA data in here.
Your readout.
Mm-hmm.
But in a high C case, it's a matrix.
Yes.
You want to project that into low dimension. How do you do the technique?
So there's actually multiple ways of doing clustering based on high C data. I think there's actually people in the audience who know this particularly well. There are ways of, there's several approaches for doing this. You can assign some, there's like a multi-dimensional scaling type approach to assign a difference. Uh-huh.
This is the every cell, the matrix.
Yes.
If you projecting cells to low dimension.
Yes.
Not necessarily.
Yes. I guess I don't totally follow. Yeah. I mean, so there's a handful of approaches that you can use. So the initial input here, right? You have the data would be 4,000 cells, and in the case of methylation, a vector of the number of bins in the genome, right? So in theory, right, you can do something similar for high C data. You could just concatenate it out into a vector. I think the other sort of logical way is you actually convert this, you do principal component analysis on the individual cell matrix, and then do clustering on the principal component. So there's a few different approaches that can be used for doing this. Yeah. I honestly, the clustering algorithm for this specific thing was done by a student in Joe Ecker's lab who was really interested in this. So I don't know exactly which of those approaches he used for this. Yeah, and so in terms of the cell types, right? So basically we can see, if we look at this gene SATB2, which is known to be a marker of excitatory neurons, we can see that this cluster in the middle basically has very low levels of SATB2 methylation. So this is what we think are the excitatory neurons, which would be this population here. FOXP2 also marks excitatory, but it's specifically deep layer excitatory neurons. So this region is hypomethylated, this is methylated. So these would be the deep layer excitatory neurons. So this is basically one way in which you can kind of reconstruct which populations and which cell types are here. And so then this is an example of that SATB2 locus. And so what we've done here is we've taken, say, all of these excitatory neurons and pooled the chromatin contact data together to generate a pseudo bulk map at this point. And so in this case, we're comparing excitatory versus the inhibitory classes. And again, SATB2, which is right here, is this well-known marker gene of excitatory neurons. And we see that there are these sort of an array of different contacts that are forming in excitatory neurons that are basically completely absent in inhibitory neurons. So we think that we can actually identify some cell type specific looping and structural events in this case. And so then one kind of just cool example of where we kind of want to go with this in the future, this isn't actually in the paper. So this is a recent schizophrenia GWAS study where they identify a handful of different loci. They can validate some of these. And so there's one that they identify that they say is a new locus they've associated with schizophrenia. It's this potential SNP right here. It's a very narrow window of linkage to equilibrium. And it's in this relatively gene-poor region of the genome. What's kind of interesting is by comparing it with the DNA methylation, and it doesn't show up well here, this is specifically in a hypomethylated intergenic region in layer 2-3 excitatory neurons, whereas it's, say, not in microglia or astrocytes. And if we look at the high C data, we can see that the high C data in layer 2-3 neurons shows actually very sort of specific long-range contacts with this CHRM3 gene that are basically absent in, say, astrocytes or microglia. So we think from this we can begin to infer things that this SNP is probably relevant in layer 2-3 excitatory neurons, and we think likely probably regulating this CHRM3 gene.
Some of this was presented at AS.
Oh, okay. Oh, cool. Okay.
So I think
Nice, cool. Okay. That's great. Yeah, so, okay, so yeah, this is basically the summary for the second part. I think I'm kind of running out of time, so I can kind of go over this quickly, that basically we've developed a method for this multi-omic profiling that we think is really interesting to allow for basically profiling 3D genome structure in vivo inside real complex tissues. And this is to acknowledge the people who did the work. This is basically my lab. Dong-Soon is the lead author on the paper that actually was just published in Nature Methods about the single cell profiling. The first part has really been a team effort between a variety of different people in the lab, including Dong-Soon and Ji Chao, as well as some technicians who have subsequently left the lab. And with that, I will take questions. Oh yeah, also collaborators, very good collaborators in terms of the methylome profiling. With that, I'll stop and take any questions.
Question?
I was wondering if you could say something about trans-interacting. I mean, cis regulatory domains are part of the story, but I think, you know, with some of the work you were doing with the MIC, you were opening the door for trans-interactions, especially with that CRISPR-Cas experiment that you were doing.
Yeah, I mean, so the MIC engineering type thing, those are still technically in cis because it's a rearranged allele. In terms of like basically, if you had a karyotypically normal cell line, trans interactions are pretty rare. That you get these kind of euchromatic, heterochromatic associations, but generally speaking, from the standpoint of like enhancer gene, it's pretty rare. There is actually some very
super enhancer.
Well, so there's very, very cool data from Stavros Lombardos's lab in olfactory neurons, where they form these giant clusters, and they actually can clearly see sort of enhancer to gene interactions between different chromosomes that seem to be critical for driving specific expression of particular. So it is out there. I'd say it's, you know, that's a very specialized cell type where that's a unique structure.
So going with the same question, so I don't know you're familiar with this notion of composite LD. You talk about linkage disequilibrium. There's another notion that you can have the LD in between gametes.
Okay.
So it's very this is introduced by Bruce Weir at University of Washington.
Okay.
And how that understanding and what you're doing.
Yeah.
Maybe you're not familiar with composite LD.
Not particularly.
Okay. Okay.
I mean, I do think that there's interesting things to think about from the standpoint of, you know, you could imagine that, you know, LD block for that SNP, right?
Yeah.
It's a very narrow window.
That's right.
The target gene is outside the block.
Yeah.
So, in some level, it actually really matters not just what you inherit there, but probably other regions. So you could imagine there might be a second SNP.
That's right.
At the locus that may be affecting the gene. So I think that it makes for potentially really complex interplay, particularly if there's multiple alleles in the population that affect expression.
It was a fascinating talk. I actually had a question about the definition of these TAD boundaries themselves. You mentioned that they're highly conserved across lineages. And I wonder, and you mentioned the loop extrusion model as well. I wonder if there are inbuilt mechanisms of how this is so highly reproducible between cell types and beyond CDCF. I mean, are there other players that are known or implicated in this definition of boundaries?
So the beyond CTCF is a bit of a, that could be a very long response. In Drosophila, the answer is yes, absolutely. In C. elegans, yes, absolutely. In mammals, it's a little bit harder. there are experiments where people have completely depleted CTCF, and you lose the vast majority of TADs and TAD boundaries. But there are actually some that are retained, or at least, partially retained. So it could be that there are additional factors that contribute to it. But I think that for the most part, CTCF is driving it. But I think in terms of the fact that these are sort of ubiquitous between cell types, CTCF is expressed like a housekeeper. So it's basically invariant in terms of expression patterns. And I think that when you look at the boundaries that we see that are very, very consistently called in different cell types, they tend to be associated with the most invariable binding sites of CTCF in the genome. So that you can have some CTCF binding sites that are unique, and those may contribute to things like cell type specific interactions. But there's clearly a subset of CTCF binding sites that just hardly ever change. And what defines that is, I don't entirely know. I think it's quite interesting. There's probably a variety of things that might contribute to it. We know that those tend to be more GC-rich regions of the genome, so it might be that they're somehow accessible just as a baseline state. But.
So in your single-cell experiment where you use a T-sneer method to cluster different cell types, so you have three kinds of feature. The first are based on methylation, and then based on Hi-C. So the result of such kind of clustering depends on how you define the dimensions. For methylation, the number of dimensions is linearly related to the length, while for Hi-C is quadratic. So how do you think the Hi-C clustering is worse, partly because there are higher dimension data?
I think the answer is yes. So I think one of the issues is kind of going back to the 1D versus 2D thing. So let's say we have, you break up the genome into 50 kilobase bins. You're going to expect a certain amount of coverage per bin from the methylome data from a one-dimensional vector, right? If you then consider the matrix, right, that same data is now being basically diluted out in that space squared, right? So it's far, far more sparse in terms of what you actually observe in the matrix compared to what you see in a one-dimensional like DNA methylation. So I think, when we pool the data together, right, you can see that there are differences. I just think that with any of those specific single contacts, you probably just don't see that really defined very frequently when you look across the different cell types. So I think that's basically what it comes down to. At least that's my guess.
Very nice talk. So when people have abolished expression of cohesin or CTCF, there's a remarkable little effect on gene expression overall. So it kind of questions the role of TADs in regulating genes. Do you think it's more to regulate enhancer gene interaction, or can you also envision that it has something to do with more of a DNA damage response, that you compartmentalize the genome into smaller units that can be used for repair and so forth?
Yeah, I think it's, so I guess there's two parts. There's clearly things that TADs seem to be doing independent from gene regulation. So they're clearly associated with DNA replication timing. I think that there's not lots, but there's growing evidence that they're associated with DNA repair. And so in that sense, I think there may be a reason why a cell has a TAD that's not from a gene expression standpoint. From the standpoint of gene regulation, like one thing that I think these activity by contact models suggest is that how a gene would respond to something like cohesin depletion is going to depend tremendously on where the enhancers are that regulate it. So that a gene that would be, say, responsive to cohesin, I would expect that it would not have any enhancers near the gene itself. Because if you deplete cohesin, you lose these kind of looping features, but the dominant kind of distance decay that you see in the data, that doesn't really change, right? So if there's a proximal enhancer in the absence of cohesin, I bet I can still find the gene and turn it on. And so at least my interpretation of those kind of results is actually that these effects of super enhancers that might actually be the most dominant feature in terms of what TADs might be doing and why they might be relevant for gene regulation, is actually to help facilitate this kind of long-range super enhancer-like contact. Because I think, these activity contact models are really interesting and cool, but that is sort of one implication, right? Is that if you're going to have an enhancer that's a million bases away, it's got to be a really strong enhancer to regulate that gene.
I just wanted to see if there was, you had CMYC there. I mean, that's a Yamanaka factor. I mean, so that's a special thing. I mean, have you looked, is there some kind of special way? I mean, you talked about its structure. Do you expect to see the other Yamanaka factors acting like that?
Yeah, I mean, so SOX2 is definitely has these really interesting ways that it uses enhancement. But that's more in the context of, like developmental regulation of the gene. So from ES cells to neural cells. Nanog and OC4, I don't quite know much about, how much they're using these kind of long-range factors.
It might be an interesting thing to look at.
Yeah. Yeah.
Because there might be some kind of special way that they're creating that, extra block. That's
Yeah.
That could be a really neat thing.
Yeah. No, I
agree. Predict that. That would be a starting hypothesis.
Yeah.
It would probably be an easy experiment. But
yeah.
So I was really interested in what you showed.
You want to?
Drove high expression being related to the overall enhancer load as opposed to, a specific super enhancer. And if there's this idea that the super enhancer is really a collection of individual transcription factor binding units that are all clustered in one place, that you're sort of seeing a similar effect when they're more distributed, but there's still a lot of them. And so I'm curious about what we can learn from high-C data in terms of the resolution to look at those individual elements. What are the limitations? Are the limitations at this point? Well, sequencing depth, it seems like we're able to overcome that if we have a big enough budget. The resolution in terms of restriction sites and maybe getting over that with something like micro C or where we're cutting more frequently. But ultimately, do you think that this technology is able to resolve interactions at a fine scale, or are enhancers inherently acting without direct contact and by recruiting things to a zone? And so the best you're ever going to get are kind of these stripes when you have clusters of enhancers in the same region.
I tend to kind of think the latter. That I actually think that if you look in high-C data, right, and you have even high-resolution data, 5 billion read kind of thing. If you see at 1 kilobase resolution that 2 sites interact, if you look 1 kilobase away, you're still going to see that they have a high interaction. So there's a strong sort of correlation in terms of the local effects that you see in a matrix. So the real question is that biological, or is that there's like some noise and some signal that's being dissipated? That's much harder to answer. I think that on some level it has to be biological, at least part of it, because the chromosome is basically a polymer, right? And so if 2 loci are close, that means the loci immediately adjacent to it is also close. So I think that feature is going to be there even if you get better and better data. Like you have micro C data and that gives you even higher resolution. It's possible that you could, maybe if you really, really sequence not just like 5 billion reads, but like 1 trillion reads, you might be able to see that. But that's getting to be crazy. I don't even know how much you'd have to spend to do that. And so in that sense, I think if you're going to find that, it would have to be you aggregate features together and accumulate. You do these metagene style.
If I may make a comment, I think that should come from combination of imaging and biochemical technique, and then you have to render mathematically how you combine these two biochemical and imaging, and then you can reconstruct a new object, and then you can render a shape to that.
I mean, I think
not the one technique alone.
The genetics, I think, are also critical, right? You have to. I think that they're hard experiments, but going in and bashing enhancers with CRISPR, you learn a lot from that kind of experiment.
Yeah.
Okay, cool.
Yeah.
Great.
Yeah.
Keep it up.
Thanks again, Jesse. Wonderful talk.
Thank you.
That's wonderful, Jesse. So this notion of composite LD. So this is I took a this is like this. So let's say this is so this is mom and dad, right? So the LD means in some sense like independence or